Hello students, today I have to start with the new chapter, that is photosynthesis in higher plants. So you know that the photosynthesis is the process of formation of organic compounds by using carbon dioxide, photosynthetic pigments, light energy. So here in this chapter I will be dealing with the details of this photosynthetic process. Now, in the previous classes, you might have studied, you might have done certain experiments which shows the essentiality of the raw material required for the photosynthesis, like the experiments to know the essentiality of chlorophyll, carbon dioxide, and light. So here I will be dealing some of the experiments which show the essentiality of all these raw materials required for photosynthesis. So let's start with it. Photosynthesis. Now in the photosynthesis, first of all, I will be starting with one of the experiment to know the essentiality of chlorophyll and light for the photosynthetic process. Yes. To explain the essentiality for these two things, here I am taking a potted plant. और ये जो potted plant है, इस plant के अंदर जो leaves present है, these are variegated leaves. So, for only here I am showing two leaves here, and these leaves, they are variegated. Variegated means some part of the leaf is green and some part is yellow. So ये जो part मैं यहाँ पर दिखा रही हूँ, this is yellow part. This one. और ये जो आस पास वाला part है, that is green. These. So yellow part, you know that in this part the photosynthetic pigment, that is chlorophyll, is absent, and this green part chlorophyll is present. अब ये जो लीफ यहाँ पर प्लांट के ऊपर प्रेजेंट है, हमने कुछ लीव्स को कवर कर देना है with the help of a black paper. So let's say that this leaf, it was covered with the help of a black paper. Right? और ये जो लीफ है, this is exposed to sunlight. अब ये जो प्लांट है, इस प्लांट को हम एलिमिनेट करेंगे. Yes, the plant is exposed to the sunlight for 1 to 2 hours. प्लांट को एलिमिनेट करने के बाद, now I will be removing these leaves. I will pluck these leaves, and these plucked leaves, they will be tested for starch. These plucked leaves, they will be tested for starch. Now, why they are tested for starch? You know this very well that in the plant, through the photosynthetic activity, there is formation of organic compounds, that is sugars, glucose is formed. Now this glucose, it is polymerized and stored in the form of starch. So it means just part of plant के अंदर photosynthesis होगा. In that part of the plant, there is formation of the product, that is starch is formed. ना स्टार्च फॉर्म हुई है कि नहीं हुई है, इसको फाइंड करने के लिए, इसको टेस्ट करने के लिए हम इन लीव्स को डिप करेंगे आयोडीन सॉल्यूशन के अंदर. Yes, so when the starch, it comes in contact with the iodine, it gives blue-black color. So starch, after coming in contact with the iodine, now it will give blue-black color. So let's see what is the scenario with respect to these two leaves when they are tested for the starch. So first see, what is the scenario with respect to the covered leaf. तो ये जो covered leaf है, जब इसको starch के साथ testing करी, जब इसकी starch के लिए testing करी, what it was observed that in none of the area the blue color it appeared. So in the covered leaf, blue color that is absent, ब्लू कलर एब्सेंट है, तो इट मीन्स यहाँ पर इस एरिया के अंदर, इस लीफ के अंदर फोटोसिंथेसिस नहीं हुआ है, फोटोसिंथेसिस नहीं हुआ है बिकॉज नाउ व्हाट इज द डिफरेंस बिटवीन दिस एंड दिस लीफ, दैट दिस लीफ इज कवर्ड विद द हेल्प ऑफ अ ब्लैक पेपर, जिसकी वजह से दिस लीफ इट वाज नॉट एबल टू गेट द सनलाइट. सो इट मीन्स वी कैन से दैट from this experiment that light is essential for the process of photosynthesis. Right? Now coming to the second leaf. When the second leaf, it was tested for starch. Then what was observed? Now this leaf which was exposed to the sunlight, when this was tested for the starch, what was observed? कि blue color appear हो रहा है, but blue color is appearing only in which area? The part which is green. Means where the chlorophyll is present, और जो yellow area है वहां पर blue color absent है. It means जो starch की formation हुई है, that is only in the presence of chlorophyll. So through this exposed leaf experiment, what is clear? That the chlorophyll is essential for photosynthesis, or the chlorophyll it is essential for the starch formation. So this experiment shows the essentiality of the chlorophyll and light. Now here I will be discussing one more experiment that will tell the essentiality of carbon dioxide as a raw material for food synthesis. So now to know the essentiality of carbon dioxide for photosynthesis, now the experiment which I will be discussing here, that is moles half leaf experiment. Now Moles, he took a plant, potted plant, and in the potted plant, in one of the leaf, the apical part of the leaf, it is placed in a bottle. It is placed in a bottle, or it is placed in a tube, and inside the tube there is presence of cotton which is soaked in KOH. तो apical part को एक टेस्ट ट्यूब के अंदर हमने प्लेस कर दिया है, जिसके अंदर प्रेजेंट है कॉटन, एंड दिस कॉटन इट इज सोक्ड विद KOH. Now it is very well known to all of you that the KOH, it can absorb carbon dioxide. Yes, it can react with the carbon dioxide. तो it means ये वाला जो पार्ट है लीफ का, इसके अंदर कार्बन डाइऑक्साइड एब्सेंट है इस एनवायरमेंट के अंदर, एंड दिस बेसल पार्ट ऑफ द लीफ द कार्बन डाइऑक्साइड इज प्रेजेंट. So in the apical part and the basal part, the environment is different, and this difference is with respect to the presence and absence of carbon dioxide. Now this plant, it was illuminated with sunlight. Yes. So for 1 to 2 hours it was provided sunlight, and after that this leaf, it was plucked, it was removed. The leaf was removed and tested for starch. And you know, for testing for starch test करने के लिए, the leaf has to be dipped in the iodine solution. When the leaf it was dipped in the iodine solution, now what it was observed? That leaf के अंदर कुछ part जो है that turns blue black. Yes, that some part of the leaf it has turned blue black. Which part it is? Basal part. Basal part, यहाँ पर जो environment था, there the carbon dioxide was present, and in the apical part, the blue color formation that is absent, blue color appearance is absent, and in this part, the environment which was present along the leaf, the carbon dioxide was absent. So it clearly shows if the carbon dioxide is available, the starch formation is present. If the carbon dioxide is unavailable, the starch formation is absent. So this simple experiment which was performed by the mole explains the essentiality of carbon dioxide as a raw material for photosynthesis. Now, after dealing with these experiments, now here I would like to discuss some of the points with respect to the historical background of photosynthesis. तो photosynthesis के अंदर कौन कौन से experiment perform हुए हैं, कौन कौन से experiment perform करे गए हैं, to know the key features of the photosynthesis by the scientists. That experiments I will be discussing here. Now first of all, in the historical background, I will be starting with the experiment of Joseph Priestley. So in the historical background, first of all, let's start with the experiments performed by Joseph Priestley. Joseph Priestley, he took a close paste, he took a bell jar, and inside the bell jar, he placed a burning candle. So this is a bell jar. And inside the bell jar, he placed a burning candle. Inside the closed jar, when he placed a burning candle, what he observed that after certain time duration, this burning candle, it gets extinguished. Right? Now later what he did, along with the burning candle, he placed a mouse here. So burning candle के साथ, he placed a mouse in this closed jar. Yes. So he placed a mouse as well as burning candle inside the closed jar, and what he's observed, after certain time duration, again the candle stopped burning and the mouse also died. Now in the next setup, what he did, again he take a closed jar. Inside the closed jar, he placed a burning candle. Again, mouse. And along with the mouse, now he placed a plant here, and the plant which was placed by the Joseph Priestley in this closed jar, that was mint plant. अब जब बर्निंग कैंडल माउस और जब प्लांट साथ में ही प्लेस कर दिया इस क्लोज जार के अंदर, नाउ व्हाट ही ऑब्जर्व दैट द कैंडल रिमेन्ड बर्निंग, स्टेड बर्निंग, एंड द माउस ऑल्सो सर्वाइव्ड. तो प्लांट प्लेस करने के बाद ये जो कैंडल है इट केप्ट ऑन बर्निंग एंड द माउस आल्सो स्टेड अलाइव्ड. माउस सर्वाइव्ड. फ्रॉम दिस व्हाट ही हाइपोथाइज्ड कि ये जो कैंडल की बर्निंग है और जब माउस रेस्पिरेशन परफॉर्म करता है, ड्यू टू ऑल दीज थिंग्स देयर इज रिलीज ऑफ डर्टी एयर इन द एनवायरमेंट. So what is hypothesized? That burning of candle and respiration performed by mouse produces foul air, dirty air, that he call it as phlogiston. Right? But this foul air, it can be purified. Yes, this air, it is purified by the plants. Because plants, they release a purified air, that is deforestation. Right? So it means burning of the candle and the respiration performed by the animals produces foul air, and the activity performed by the plants purify this air. तो जो foul air है उसको क्या कहा? phlogiston. और जो purify air है उसको क्या कहा? dephlogiston. And actually what is this phlogiston? This is the carbon dioxide which is released through the respiratory activity of the mouse, and dephlogiston is oxygen which is produced by the plants. Yes, plants, what they will do? They will consume this carbon dioxide, photosynthesis perform karenge, and you know that one of the byproduct which is formed in the photosynthesis, that is oxygen, which results in the dephlogiston. So dephlogiston is oxygen and the phlogiston is carbon dioxide. Now the dephlogiston, it was discovered by the Joseph and Priestley in 1774. So this point is very much clear to you. Now after the Joseph Priestley, now I will be discussing the contribution of engine house. Now Jane engine house. Now he used a similar setup which was used by Joseph Priestley. तो सिमिलर टाइप ऑफ दी सेटअप इट वाज यूज्ड बाय इंजन हाउस. और ये जो सेटअप है इसको इसने प्लेस करा वंस इन द डार्क एंड वंस इन द लाइट. एंड व्हाट ही ऑब्जर्व्ड दैट अ प्लांट कैन प्यूरिफाई एयर only when the light is present. Yes, only in the presence of light plants they can purify air. It means that the process which is involved in the purification of air, that process requires light. So one of the essentiality for that process is light. So this is how Jane engine house showed that the light is essential for the process which purifies air. Along with this, he also performed one very famous experiment that was with respect to water plant. He took a beaker Inside the beaker, he took a water plant, like hydrilla, and over the water plant he placed a inverted funnel. Yes, so inside the beaker there is water plant, and over the water plant he placed a inverted funnel, and he illuminated this setup by the light. He illuminated it, right? What he observed, that in the presence of the light, the green parts of the plant, they produced bubbles. जो ग्रीन पार्ट्स है प्लांट के, उसके आसपास बबल्स प्रोड्यूस्ड हो रहे हैं. Now later on, these bubbles, they were tested, and in the bubbles the gas which was observed, that is oxygen. So through this experiment, what he concluded, only green parts of the plant, they produce purify air, that is oxygen. So green parts of plants, only they have the ability to produce oxygen. Now after the engine house, let's see what is the contribution of Julius von Sack. Now Julius von Sack, what he found, that the plants, they have the ability to form glucose. And this glucose, it can only be formed by green parts of plant. And this glucose, it is mainly stored in the green parts of the plant in the form of starch. So this is very much clear. Jan Ingenhousz, he showed that the green parts of the plant, they can release oxygen, they can purify air, and Julius von Sack, they showed that only the green parts of the plant, they can form glucose, which is mainly stored in the form of starch. Now in the middle of the 19th century, the key elements which are involved in the photosynthesis, they were almost known, and the empirical equation of the photosynthesis was given at that time. So now what is that empirical equation for the total photosynthesis process that I will be writing here? So empirical equation of photosynthesis, which was given earlier in the mid of 19th century. That is, carbon dioxide and water in the presence of light results in the formation of carbohydrate. Formation of carbohydrate, and one of the byproduct which is produced, that is oxygen. Now according to the earlier view कि जो photosynthesis के time पर oxygen release होती है, that is released due to the breakdown of carbon dioxide. But today we know that the photosynthetic oxygen is not contributed by the carbon dioxide, it is contributed due to the splitting of water. So now here I will be dealing with the three experiments. I will be discussing three experiments here which explain this, which showed that the photosynthetic oxygen, it is contributed by water rather than the carbon dioxide. So let's discuss three experiments which showed that photosynthetic oxygen, it is contributed by water rather than the carbon dioxide. So here I will be discussing experiment which was performed by the Van Niel. This is a milestone contribution towards the understanding of photosynthesis. अब वेल नील ने जिस फोटोसिंथेटिक ऑर्गेनिजम्स को यूटिलाइज किया, दैट इज बैक्टीरिया. एंड विच बैक्टीरिया? दैट इज sulfur bacteria, like purple sulfur bacteria, green sulfur bacteria. Now the next one, the experiment performed by the Robert Hill, and now the experimental material which was utilized by the Robert Hill, that is angiospermic plant, that is Stellaria. Next one, the experiment performed by Rubin and Kamen, and in this experiment the unicellular green alga was taken, and that unicellular green alga represents chlorella. So let's start with the contribution of Van Niel. Now Van Niel, he took sulfur bacteria, and what he observed that in the sulfur bacteria for carbon dioxide fixation, instead of the water, there is utilization of sulfurous compound, one sulfurous compound, that is H2S. According to the old view, ऑक्सीजन जो रिलीज होती है फोटोसिंथेसिस के अंदर, that is due to the splitting of carbon dioxide. Now in this case the carbon dioxide utilization is occurring, but in this bacteria the oxygen release is absent. Oxygen release is absent, it means they perform an oxygenic photosynthesis. So in spite of the utilization of the carbon dioxide, in this bacteria during the photosynthesis, the oxygen release is absent, which showed that photosynthetic activity के time पर carbon dioxide की splitting नहीं होगी. Instead of that, there is breakdown of H2S. Yes, H2S का breakdown होगा. H2S का breakdown होगा. The hydrogen is utilized for the reduction of carbon dioxide to form the carbohydrates, and the sulfur is released as a byproduct along with the water. So instead of the carbon dioxide, there is a splitting of H2S, which results in the formation of carbohydrate. Along with that is production of elemental sulfur and water. So this is the equation of the photosynthetic in this anoxygenic bacteria. Now he compared the bacterial photosynthesis with the plant photosynthesis. In the plant photosynthesis, carbon dioxide fixation के लिए क्या use होता है? Water use होता है. So again, what he concluded that instead of the splitting of the carbon dioxide in the green plants, the splitting of water. Splitting of water, which results in the production of oxygen. So in the green plants, now the equation can be written as 6 carbon dioxide, 12 water molecules in the presence of light results in the formation of glucose. You know that the glucose is a 6 कार्बन शुगर. तो ये जो 6 कार्बन डाइऑक्साइड है फिक्स होने के बाद बनाएंगे 6 कार्बन शुगर, that is glucose. Along with that there is release of the 6 water molecules as well as there is release of oxygen, right? So this is the photosynthetic equation for the sulfur bacteria, and this one is for the green plants, which clearly showed that the photosynthetic oxygen, it is not contributed by the carbon dioxide. This is very much clear to all of you. Now here I would like to add one point that जो prokaryotic organism है, जो photosynthetic bacteria है, they perform an oxygenic photosynthesis, that the oxygen release is absent. But do you know any bacteria which perform oxygenic photosynthesis? Yes, I had discussed this in the previous chapters. That bacteria is cyanobacteria, that is BGA. So you please note this down here, that the bacteria which perform oxygenic photosynthesis, that releases oxygen during photosynthesis, that is cyanobacteria. And one example of the cyanobacteria I'm writing here, that is the spirulina. Right? अब ये जो बैक्टीरिया ये परफॉर्म करते हैं एनऑक्सीजेनिक फोटोसिंथेसिस, एंड ग्रीन प्लांट्स एंड बीजीए दे परफॉर्म ऑक्सीजेनिक फोटोसिंथेसिस. नाउ व्हाई इट इज सो, दैट आई विल बी डिस्कस इन द फोर्थ कमिंग पार्ट ऑफ द चैप्टर. So now let's see the contribution of Robert Hill. In a beaker, Robert Hill took chloroplast of Stellaria so in the water medium, he took chloroplast of angiospermic plant. Just now I have told you this, and that angiospermic plant, that is Stellaria. So this medium contains chloroplast of Stellaria. Now in this medium, now the water is present, but you have to keep this in mind that carbon dioxide is absent. This medium is carbon dioxide free, and what he did, he illuminated this setup. Right? He provided light to this setup. अब इस मीडियम के अंदर वाटर प्रेजेंट है और कार्बन डाइऑक्साइड एब्सेंट है. And according to the old view, if the oxygen is releasing in the photosynthesis, that is due to the breakdown of carbon dioxide. But in this medium, the carbon dioxide is absent. And if the old view is correct, the carbon dioxide is absent, it means the oxygen release should also be absent. But what Robert Hill observed, जैसे इस setup को light provide करी, that in spite of the absence of the carbon dioxide, the oxygen was released by the chloroplast. So this showed कि कार्बन डाइऑक्साइड की एब्सेंस के अंदर भी ऑक्सीजन का रिलीज हो रहा है. So it means instead of the carbon dioxide, the oxygen contributor is water. Now what he did, to know this, in this medium, he placed a hydrogen acceptor. To know this, in the medium, he placed a hydrogen acceptor. So the hydrogen acceptors which can be placed, one, that is benzoquinone, then chromates, ferricyanides, and DCPIP. DCPIP, dichlorophenol indophenol. So let's discuss DCPIP here. तो इस मीडियम के अंदर उसने प्लेस करा DCPIP. Now DCPIP, it exists in two states. In the oxidized state, it is blue in color. And in the reduced state, that is after accepting the electrons, it changed into colorless. Yes, it gives the colorless state, right? अब यहाँ पर रॉबर्ट हिल ने DCPIP को प्लेस करा इन दी ऑक्सीडाइड स्टेट. ऑक्सीडाइड स्टेज के अंदर प्लेस करा, इसका मतलब ये जितना भी सोल्यूशन यहाँ पर प्रेजेंट है ये सारा ब्लू इन कलर है. द होल ऑफ द सोल्यूशन इज ब्लू इन कलर, राइट? सो ही प्लेस्ड DCPIP in the beaker in the blue state. और जैसे लाइट प्रोवाइड करी, लाइट प्रोवाइड करने की वजह से वाटर की स्प्लिटिंग हुई, वाटर की स्प्लिटिंग से रिलीज होंगे इलेक्ट्रॉन्स, हाइड्रोजन आयंस and oxygen, right? The electrons, the hydrogen ion, they are accepted by the DCPIP because it is a hydrogen acceptor. और जैसे ये reduced होगा, तो reduced होने की वजह से now the blue color is lost and the solution in the suspension, it will turn colorless. So, पहले कौन से कलर का सोल्यूशन होगा? ब्लू कलर का. बट आफ्टर द स्प्लिटिंग ऑफ द वाटर, आफ्टर एक्सेप्टिंग द इलेक्ट्रॉन्स एंड हाइड्रोजन, द डीसीपीआईपी इट इज कन्वर्टेड इनटू द कलरलेस रिड्यूस्ड स्टेट. अब ये जो हाइड्रोजन एक्सेप्टर डीसीपीआईपी, यहां पर रॉबर्ट हिल ने जो यूज करा, दीज हाइड्रोजन एक्सेप्टर्स दे आर नोन एज हिल oxidants. So it's very well known to you what is an oxidant, जो खुद reduce होगा और दूसरे compound को oxidize करेगा, right? So all these compounds they are known as Hill oxidants. अब यहाँ पर जितने भी मैंने oxidants का नाम लिखा है, they are all artificial, right? Can you tell me in the chloroplast which is the natural Hill oxidant जो इन electron hydrogen ion को accept करता है? The natural Hill oxidant that is NADP plus. After accepting the electrons and hydrogen ion, it is converted into the NADPH2. So after accepting the electrons and hydrogen ion, it is converted into the NADPH2. So these are the artificial Hill oxidants. This is natural Hill oxidant present in the chloroplast, and you have to keep this in mind that this natural Hill oxidant, it was not discovered by the scientist Hill, it was discovered by the two scientists, that is Virchow and Ochoa. So after dealing with the Hill experiment, now I will be starting with the next one, which was performed by Rubin and Kamen. So now the experiment performed by Rubin and Kamen. Now Rubin and Kamen, they took chlorella, that is a unicellular green alga. Suspension of chlorella was taken in water, right? And in this water, the oxygen which is present, that is radioactive oxygen. That is, instead of the normal oxygen, there is presence of the O18 oxygen. But the carbon dioxide which is present in the medium, uske andar jo oxygen present hai, that is the normal oxygen, that is non-radioactive form of the oxygen, that is O16. तो वाटर के अंदर प्रेजेंट है रेडियो एक्टिव ऑक्सीजन और कार्बन डाइऑक्साइड के अंदर प्रेजेंट है नॉन रेडियो एक्टिव फॉर्म. और ये जो सस्पेंशन है इस सेटअप को उसने लाइट प्रोवाइड करी. सो जैसे लाइट प्रोवाइड करी, देन देयर वाज रिलीज ऑफ ऑक्सीजन. And this oxygen, it was tested, and what it was observed, that instead of the normal oxygen, the oxygen which is produced, this is radioactive, which clearly showed that in the photosynthesis, the oxygen which is released, that is not contributed by the carbon dioxide, the contributor is water. So all these three experiments with respect to the photosynthesis, it is very much clear to all of you. Now after this, now I will be starting with the site of photosynthesis. So where the photosynthesis it is performed in the plant.